All right. Good morning, good afternoon, good evening, wherever you are in the world. Thank you so much for joining us. Welcome to our webinar. Today, we are Arima Genomics, and today we'll be talking about achieving chromosome-scale assemblies with Arima Hi-C. I just want to give you a brief outline of what we're going to be talking about. So the first thing is going to give you an overview of our 3D genome technology. Then we'll talk about the benefits of this technology in genome assembly applications. Then I'll do a deep dive into our Arima high coverage Hi-C solution, which is specifically designed for genome assembly. And then we'll wrap it up with conclusion and Q&A. My hope is that we convey who we are and what we do, showcase how Hi-C data is critical for building chromosome-scale genome assemblies, and to prove why the Arima high coverage Hi-C technology is the best solution for your genome assembly projects. I also wanted to mention we have an ongoing grant program that I'm going to have more information about at the end of this presentation. So stick around and you'll see some information on that as well. Just to kick things off, just wanted to give an overview of our company. So Arima Genomics provides kits and reagents to help enable our workflows in your lab. We've been commercializing our technology since for the past 5 years or so. But we also provide services and support to enable customers to send their samples to us, and we can process and perform our workflows in-house as well. So we are a full end-to-end service provider. And you know, some of the things that people love about working with Arima is that our chemistry has a flexible sample input type with both the types of samples that we work with and the input requirements that we're compatible with. Our data is compatible with the latest assembly and scaffolding pipelines. And we really have a science-first approach. You know, we're a company that we're very easy to partner with, and we are very invested in making sure that your projects are successful. So now I'm just going to give a background on the Arima 3D genome workflow. So our technology is powered by Hi-C, which stands for high throughput chromatin conformation capture. And this chemistry reveals how spatial and temporal changes in chromatin conformation alter gene regulation and cellular function. And the way this technology works is just why this technology is important is that, you know, sequences are linear, but DNA is not. And so we want to capture that 3D genome information, which does reveal a lot more insights into the into the biology of your sample. So the DNA is compacted into nucleus, and so what our chemistry does, the goal of our chemistry is to extract the DNA while preserving the conformation data. So we cross-link the DNA, we cut and religate that DNA, and then after fragmentation sequencing, the results are the ability to explore 3D interactions and to discover the power of 3D genomics. So again, 3D, this is useful in gene regulation, understanding cell types and diversity, and human health, and genome assembly. Ultimately, with our chemistry and technology, you gain unparalleled access to the sequence, structure, and regulatory landscape of any genome. So, you know, how is Hi-C specifically used in genome assembly? As we all know, over the past 2 years, sequencing technologies have evolved quite a bit. Reads are getting longer and they're getting higher quality. So now when the typical lab is doing a genome assembly project, they use long read sequencing data to assemble that, which results in hundreds to thousands of unmapped contigs. Now, while the reads are long and high quality, you're still missing the context of that read, or in Arima Hi-C provides that context that's used to scaffold contigs into their location in the genome, and also enables anchoring those contigs into a complete genome assembly. And this leads to biological insights, which inform conservation, agriculture, and human health. But ultimately, which we'll cover in the following slides, is you can't get phased chromosome-scale genome assemblies with just sequencing alone. And here's how Hi-C helps get those, kind of resolves the gaps with existing technologies. On the left-hand side is the scaffolding. The Hi-C technology is used to order and orient contigs, and also to anchor contigs into their actual chromosomes. Furthermore, after scaffolding full assembly, the same Hi-C data is used to fix misassemblies or potentially identify structural variations depending on the application. If you're comparing two genomes against each other, this could be a useful tool to kind of determine what the structural variations are. And then finally, Hi-C data can be used to phase haplotypes, which I'll cover in the next slides. So the first step is in a genome assembly project is to order and orient contigs. Current long-read technologies provide high-quality contigs and long ones, but they do not provide the context. And the challenge with this is that in complex genomes, particularly ones with pseudochromosomes or large numbers of repeats, structural variations, we may not get a correctly ordered or assembled assembly. And so in this example that I'm showing here, researchers at the University of Saskatchewan are comparing differences between cultivated lentils versus their wild relatives. And large-scale structural variations, including inversions and copy number alterations, exist between the two groups. And so these variations make it difficult to, you don't have a common reference that can be compared against because of these variations. And so the solution that we provide is that chromatin structures delivered by Arima HiC data provide DNA proximity information. The interaction probabilities are used to order and orient the contigs. And in this example, again, researchers used HiC data to order and orient contigs and generated for the two lentil groups, resulting in two reference assemblies, one for the cultivated and one for the wild lentil species. So the takeaway is that even as reads get longer and improve in quality, without the 3D context, the orientation and ordering of these contigs are not possible. Once you have ordered and oriented the contigs, the next step is to anchor them into the full chromosome. The problem is that once contigs are ordered, the resulting scaffolds need to be assembled, and generating end-to-end assembled chromosomes with long reads alone would require significant genome coverage, which drives up the project costs without necessarily improving the build quality. And also, despite advances in read length and quality, very large repetitive regions like those found in many plant genomes and in telomeres are still often too long and repetitive for current long-read sequencing technologies to completely sequence through. So HiC overcomes these challenges by providing long-range contiguity information. And the data combines the benefits of cost-effective, high-quality short reads and the positional sequence context of HiC with the high-quality long reads to have a fully assembled and complete genome. And in this example, researchers at the Chinese Academy of Agricultural Sciences, they use HiC data along with optical maps to help anchor contigs into the 7 chromosomes for the cucumber genome. Anchoring contigs to chromosomes is more cost-effective, accurate, and efficient when supplementing long-read data with HiC. Now we'll talk about fixing misassemblies. In addition to the challenges I posed earlier around scaffolding, the complexities associated with highly repetitive regions and genomes exist across multiple species. And these repeats can result in misassemblies. And in this example, the hummingbird genome, hummingbird genome example from the Vertebrate Genomes Project, there's a misassembly that exists here on this left-hand side. You can see that there's a misjoin indicated by this red arrow. The HiC data provides 3D context, and this in turn delivers information around the chromosome territories thanks to the principles of chromosomes primarily interacting with itself. So you can see in the same plot how this supposed single chromosome actually has HiC interactions, which clearly indicate that this is not one chromosome, but actually 2. And so the corrected genome assembly was easy to perform visually through the HiC data and allows for a more curated and accurate, fully constructed genome in this example. So the takeaway here is that HiC data provides 3D context, which enables error correction of genome assemblies. And this is especially useful for highly repetitive regions in genomes across multiple species. And finally, an exciting area is around phasing haplotypes. As long-read technologies continue to have higher quality, the SNVs found in each contig can indicate how 2 contigs from the same chromosome belong to separate alleles. However, without knowing how contigs relate to each other, it is not possible to fully assemble completely phase these haplotypes. This can be resolved when you have parental sequence data, at least in mammalian or eukaryotic organisms. However, this is often either not available or often not cost effective. And then, other challenges occur when organisms are multiploidy like plants, which presents an even larger challenge. And so the 3D genome data provides context in addition to allele-specific to those allele-specific variants by connecting contigs from the same allele with each other. And so the 3D genome data provides allele-specific context and to scaffold contigs that are from the same allele, resulting in a phased assembly. So, we talked a lot about the benefits of Hi-C to genome assembly, and I wanted to now introduce how Arima Hi-C has developed an optimized solution for genome assembly applications. So I'm going to introduce the Arima high coverage Hi-C workflow. Many researchers prepare their own Hi-C reagents. And the challenge with that is that the chemistry can be difficult to build. It can vary dramatically in performance. And from lab to lab, it might be difficult to replicate the results when labs are just preparing their reagents on their own. So, it's important to work with standardized reagents that work consistently and reliably across multiple types of species, because it could be replicated easily in the future. So we developed the Arima high coverage Hi-C kit in partnership with leading genome assembly consortia. The chemistry is designed to work consistently across multiple sample types and inputs, and designed to provide reliable data quality across different organisms and species. Our kits come in 8 and 48 reaction kits, so it's flexible depending on what the volume of samples that you're working with are. And the protocols are user-friendly, and we provide robust and reliable results, which I'll show in this next slides. So I think it's important to understand why we built this chemistry. And so to develop a robust and reliable Hi-C kit, we partnered with VGP and the Darwin Tree of Life to better understand what would actually make a good chemistry. And so here I'm showing 3 charts. The first chart is uniformity plot showing normalized sequence coverage on the x-axis and the percent of bases sample in the y-axis. On the right-hand side, the second and third charts are in silico models generated using VGP genome data showing percent genome coverage on the y-axis here. And so the challenge that we're trying to overcome is to enable these groups to build high-quality, cost-effective chromosome-scale assemblies. But the problem with some of these legacy-based approaches is poor uniformity, which can result in poor sequence coverage. And so in this example here, this legacy Hi-C chemistry, which we sourced from the Rao et al. Paper, indicates how this poor uniformity that is generated from a single enzyme chemistry results in poor sequence coverage and inconsistent performance across different organisms. Even our flagship dual enzyme chemistry, which we use in a lot of our gene regulation applications and structural variant applications, while it has much better uniformity, we still didn't quite achieve, consistent sequence coverage across all these different genomes. Plus, there were organism differences that were driven by sequence composition changes. So we built our Arima high coverage Hi-C chemistry to provide very high uniformity and consistent sequence coverage performance across multiple genomes. And we are showing here that, not just in that the performance is consistent regardless of the organism that we're working with. So, the takeaway is that, with this 4-enzyme chemistry, you're going to get a much higher, more uniform coverage across your genome, which results in better assemblies. And another useful application of Hi-C data in genome assembly, again, is going back to phasing. And for phasing, we rely on SMVs to distinguish alleles from each other. And just to kind of pressure test the reliability of our chemistry versus gold standard whole genome sequencing, we wanted to just see, can our chemistry reproduce results similar to standard short read sequencing data? And what we're showing here on this left-hand graph is our SNV sensitivity, which is across 4 human genomes on this x-axis with different coverage ranges. We're reaching about 95 to 97% SNV sensitivity compared to whole genome sequencing. And the concordance is also extremely high, with greater than 99.7% concordance with whole genome sequencing methods against a reference genome. So, with this, we are providing not just consistent sequence coverage, but the actual sequences themselves are actually very reliable and providing really good high quality information about your sample. So, most who do their homework know that there are other methods for Hi-C, particularly DNA-spaced method. And so we wanted to provide some information how we stack up against a DNA-spaced version of Hi-C. So just as some background, we obtained data from publicly available human pangenome reference consortia. In this data set, the 37, we have a comparison of Arima versus DNA-spaced Hi-C, one set of data at 37x coverage and another set at 70x coverage, just to kind of compare the performance of the two. The Arima data is from 2 separate libraries. And then the 37x coverage data, DNA-spaced, is from 2 libraries to produce this 37x, and then 3 libraries to produce the 70x data. So some of the things that we observed in this comparison is the first round of going from raw depth to mappable reads. We're already seeing some degradation of reads resulting from unmappable reads. And then second round is PCR duplicates were significantly higher with a DNA-spaced method versus our Arima Hi-C library prep method. And then this results in about 20 to 22% fewer map dedupe reads with the DNA-spaced methods, or roughly 20 to 30% more usable reads using Arima high coverage Hi-C methods. So at the same depth, Arima yields more usable data. But again, sequencing technologies are evolving, reads are becoming cheaper. So, perhaps the read depth issues associated with DNA-spaced Hi-C maybe not be as big of a concern to some people. However, it's not just about the amount of usable reads, but the quality of those reads. So on the left-hand side, we break down the number of mapped reads into trans, short-range cis, and long-range cis. And same for Arima Hi-C versus a DNA-spaced Hi-C. But when you break that down into percent of mapped reads, you can see that the DNA-spaced Hi-C methods produce significantly more trans signal and a lot less long-range cis signal. The problem with this is that trans signal is interchromosomal reads. And this becomes an issue because the principles that we rely on are that chromosomes primarily interact with itself, and that helps you reliably scaffold and orient the contigs and generate full-scale assemblies. But when there's a lot of trans signal in your data, especially when there's no reference established where it's unclear what is trans and what is cis, the reliability of your output becomes questionable. And so, the Arima Hi-C data consistently provides low trans signal, but a large amount of high quality cis, particularly long range information. In addition to that, we also wanted to look at, what are some of the other issues associated with regard to sequencing errors generated by high coverage Hi-C versus DNA-spaced Hi-C. One of our observations is that our SNV sensitivity at lower depths is a lot higher compared to a DNA-spaced method. And then in addition to that, there's the issue with false positives. So we have about 0.2% false positive rate versus the DNA-spaced method as 1% false positive rate. And so we're seeing a reliability of the actual sequence data being compromised when using a DNA-spaced method. And this can be compensated if you double the amount of reads, but then that's a significant cost burden just to kind of compensate for poor quality data. And so the result ultimately becomes poor quality data impacts assembly performance. And in this example here, we started with 30.8 with the initial assembly with 30.8 MB pre-scaffolding N50 quality. And post scaffolding, we have 131 megabases of N50 data. So high coverage, Hi-C uniformity mixed with superior quality results in a really strong overall assembly, versus the DNA-spaced method, which used this under the same conditions, results in 104 megabases of high quality data. So lower Hi-C data quality and unmappable reads results in lower quality scaffolds in assembly. And so these are the reasons why, Arima has developed successful partnerships with large consortia like the VGP, the European Reference Genome Atlas, and the Wellcome Sanger Institute, or the Darwin Tree of Life project. And there are more than 200 genome assembly publications and counting that are leveraging Arima high coverage Hi-C chemistry to supplement their long read sequencing for their assembly projects. And these genomes are a strong foundation for diverse areas of research, from agriculture, sex chromosomes, unraveling sex chromosomes, evolution and determination, and conservation, translational research, and human genomics. And so with that, I'll lead to with these conclusions that you can't get phased chromosome-scale genome assemblies with sequencing alone, because sequence context is required for orienting and ordering contigs, correcting and scaffolding errors caused by long repeats, anchoring contigs to chromosomes, and phasing haplotypes. And Arima high coverage Hi-C addresses these issues by providing enhanced uniformity, coverage through our 4-enzyme chemistry, consistent performance across species and sample types, high quality 3D genome data, and then providing more usable high quality reads. And ultimately, we efficiently generate high quality chromosome scale assemblies across multiple species. And if you want to learn more about how Hi-C can be used for genome assembly, we just launched a fantastic new ebook. Feel free to go ahead and scan this QR code here. I also want to encourage you to take a look at one of our earlier webinars from last year, where we had a panel discussion with Dr. Giulio Fermenti at the Rockefeller University, Dr. Shilpa Lagarde at University of Copenhagen, and Dr. Alex McIntosh at University of Edinburgh. These are phenomenal scientists where, my colleague, Pamela Bentley Mills, hosted this webinar, and it was an incredible discussion covering not just techniques, but also computational methods. So there's a lot of insights to be gained from this talk. And finally, I also wanted to mention our 2023 Multi-Omics Grant Program. Now, this is a genome assembly webinar, but this grant program is an exciting opportunity for anybody who wants to get started with using one of our kits. So the requirements: submit a 200-word proposal by March 3rd, 2023. We will select 2 recipients who will be rewarded with a Hi-C kit and a library prep bundle of their choosing. And it's a great opportunity, regardless of where you are in your career stage or where you are with your level of experience, to get your hands on this on this chemistry and technology. Feel free to scan the QR code or visit arimagenomics.com/grant. And with that, I have concluded the presentation, and I wanted to open it up for Q&A. And Dr. Kristen Sikink will join us for that. The first question is, does DMSO preservation of the sample interfere with the Hi-C protocol?
Yeah, so
go ahead.
I can take that. I would say if we're talking about something like a DMSO based cryopreservation media, it's probably going to be fine. Our preference would probably be for flash frozen tissue. The first step is formalin fixation. So if you have fresh or flash frozen tissue, that's probably going to be better, but should probably be okay.
All right. Thank you. The next question is from Dr. John Birmingham. How do you tell when your Hi-C heat map plot is fully curated? So do you want to answer that, Kristen, or should I try to
go for it?
So
you've got the pictures.
Yeah, so the Hi-C heat map, I can go back to this slide here and try to zoom in on this. This is an example scaffolded assembly from a Hi-C heat map. And actually, this is not a curated heat map. And you can see in this plot here, there are red signals that are off the diagonal here. These are actually examples of misassembly. And so this data was generated using 3D DNA as that pipeline, and that pipeline allows you to kind of move contigs around and to just correct your assemblies, visually. And then more modern pipelines, I believe, are actually doing that in an automated fashion. So the Hi-C data visually provides that signal that lets you know that the assembly, there's errors in that assembly. And then the curation process is just basically correcting those errors and trying to minimize the off-diagonal signal as much as possible. Dr. Nadia, can 4-enzymes tech help capture AT-rich regions? So yes, we are the chemistry that we built was tested across multiple different types of sequence compositions. Certainly, different organisms would have different levels of uniformity, but in connection with feedback from our colleagues at VGP and Darwin Tree of Life, we're still getting really rich coverage across the genome. But there will be some regions that will be more challenging to cut through. But the three-dimensional information helps you kind of resolve around that. Right. Dr. Brunhara, when interested in structural variation across many samples, are the recommendations the same as those for de novo genome assembly in terms of coverage, library prep, etc.? I'm wondering how it could cut costs and analyze multiple samples. Kristen, any thoughts there?
Yeah, so as long as you already have a good reference genome, I would say that for structural variation in particular, if you're looking for large translocations, things like that, you probably don't need as high a coverage from the sequencing. I think the sequencing depth that we've had good success with for that is probably 150 to 200 million reads for like a human-sized genome. Whereas for the de novo assembly, you might need something more like 600 million. So I think in terms of the library prep, the recommendations are going to be very similar to the genome assembly, HiC, all of that's going to be pretty similar. But for sequencing coverage, you could probably get away with less for those types of questions. Depends on the structural variation that you're looking for, though. Smaller rearrangements on the same chromosome, you might need higher depth of coverage to get to those, but bigger interchromosomal translocations, maybe you can see with less. So,
thank you. Yeah, and I guess I should mention that we do have software. We work with an open source software called HiC Breakfinder, and while our workflow for structural variant applications is optimized for human genomes, once you have a reference assembly, their pipelines allow for uploading an expectation file. And if you have a reference, you can just compare against that expectation file, and allows for structural variant detection just using HiC data alone, potentially. Yep. Okay. And any specific software for scaffolding or open source software like Salsa, et cetera, will work. And yes, so open source software will work. I think we've most routinely internally used Salsa. Our ebook has a lot of information about all the different software and tools that are available and what are some of the kind of advantages and uniqueness that these different tools provide. Okay, yeah, this is a good question. How do you determine the cis and trans reads for the non-model polyploid plant samples? Kristen, I'll take a stab at this, and then you can kind of fill in the gaps from there. But the cis and trans is determined once you have established a reference. So once you build your reference genome, then you can kind of go back to your HiC data and then determine how much of that signal is cis versus trans. So in human genomes, where it's a very well established, when we develop our chemistry, because it's a well established and known genome, we can characterize the properties of our chemistry and measure cis and trans rates. But when you're working with de novo assemblies for non-model organisms, you have to rely on the reliability of the chemistry first. You build your reference, and then you can kind of measure cis and trans rates after that. Did I capture that correctly, Kristen?
I think you did good.
All right, next question. Dr. Tiagi, do we need biological replicates for Hi-C? Do those help in removing false TADs? Kristen, you want to take this one?
Sure. I think replication is always good. It's always going to help you. And being able to, have replication of TADs is never a bad thing. That being said, they're not absolutely necessary unless you're specifically looking for TADs. In particular, you're going to get a lot of signal that for like assembly, I think you're going to be okay without necessarily having a huge number of replicants, but replication is always good. I'm never going to say no replication.
Thank you. Dr. Farhan, has this been tested for Drosophila genome, and how much depth read do you suggest for Drosophila genome given its small size? I personally don't know off the top of my head. I know that we have, more than 200 genome assembly publications using Hi-C, and I do believe we have a publication for Drosophila. So I think, Dr. Farhan, if you reach out to us, we can send you that publication with what experimental parameters they use to assemble that genome. And then if we'll get back to you with that one, with the specific information there. What is your preferred way of generating contigs for scaffolding? So historically, and Kristen, tell me if you can kind of step in here as well, but I think historically we've been using salsa, but there are some newer technologies and newer methods that have been published, which seem quite compelling. So I think our team is actually looking into that. The tried and true method is salsa for scaffolding, but then I would encourage kind of reviewing some of the other options that were recently published, even within the past year.
Yeah, I agree with that.
And then another question around SVs. So what software do we recommend to capture SV? So as I mentioned, there's Hi-C Breakfinder. That's a software that was developed by Jesse Dixon and Feng Yue in Northwestern. Jesse is now I believe at UCSD. And then also Dr. Yue has another method called NeoLoop Finder, which you might want to reach out to them about as well. We use Hi-C Breakfinder for our human genome SV detection, and then the NeoLoop Finder seems to be something that it's a fairly newer method and might be more flexible for non-human genomes.
I think the NeoLoop Finder may also be more flexible for different types of structural variation. Right. Hi-C Breakfinder works really well for translocations and things, but not so great for finding copy number variation and stuff like that. Hi-C Breakfinder for us is part of the Arima SV pipeline that does kind of that structural variation detection.
Right. That's right. So how does chromosome number affect scaffolding with Hi-C? For example, is a large genome with many chromosomes easier than a large genome with few chromosomes?
So I think that is something that'll kind of be dependent on the structure of that genome and kind of the regions in that assembly, how much repetitive stuff you have in there. Like there's going to be other factors, I guess, that go into that beyond just how many chromosomes, and I think that's probably going to affect your difficulty in assembly more.
That's right, and I believe if I'm it's probably more about the composition of those chromosomes rather than the size.
Right. Yeah.
Because if that very large chromosome consists of very distinct regions with very few repeats, it actually might be easier than a organism with small chromosomes that are almost all repeats. And so what the data helps in conjunction with long read data, the long read data is useful for kind of trying to push through as much of that repeat regions as possible with a single contig, but then we can help kind of orient those contigs to the other ones to kind of make a more complete assembly. So I think it's it's really a question of like, in the experimental design, we want to make sure, particularly for larger organisms and larger genomes, to leverage both the long read and the Hi-C technology because they both they complement each other very well. Is the difference between using Hi-C for genome assembly and TAD IDing simply in program settings? On my end, I think that's so. Unfortunately, I don't know the answer to that question. It's more of a technical software question that we would have to follow up with you on, so we can get back to you on that question as well. We'll reach out to you and make sure we get you some answers from our bioinformatics team. What is your experience with samples preserved and RNA later frozen? Can the Hi-C protocol work for this type of tissue?
I haven't personally done anything with RNA later preserved samples. It would be worth trying. I'm not sure if anyone here has experience with that, though. We could look into that and get back to you.
Right. And I think that if I'm not mistaken, there is a publication put out by VGP on sample preservation methods that we can link out to our to the attendees here as well. So I think they have a few methods that they compared that showed what's the best way to collect and preserve your samples. What is the minimum contig and 50 required for Arima Hi-C scaffolding? I think we're pushing, at least for me, I'm pushing the boundaries of being able to answer that accurately and reliably. So I think we'd have to go back to you, Kristen, or maybe bring that back to our tech support team.
I think we'd bring that to the tech support team and our bioinformaticians and ask them that question.
Thank you so much. Yeah. All right. Another bioinformatics question. What is the best bioinformatics pipeline to map Hi-C reads and produce a scaffolded genome? I did try mapping my OmniC reads to HiFiASM produced assembly and used Salsa for scaffolding, but the assembly did not approve. Any thoughts? And I think that's, again, I think that's another example of, we should probably set up a phone call and let's go through the data together and just understand what, what happened there. There are newer tools that exist. I think HiFiASM is actually a pretty new tool that's out there. So I think we'd have to kind of dig into the data itself and see if there's an issue even with the source data. Again, that's a DNA based method, and it might produce its own artifacts that might be problematic. So we just have to kind of see what the data looks like and then go from there. The juicer pipeline you recommend for analysis is tedious to set up, especially for computational resources, CPUs, GPUs, MEM available to different users. Is there an easier and equally good alternative? So this is about visualization, right? And then I think there's open source online visualization tools, right? Kristen?
Yeah, so juicer would be what we would use to produce the heat maps that we're visualizing here. Juicer is really my go-to for this. So I think there are potentially some alternatives, but I don't have experience with them to know if they're easier or better.
Yeah, and I think maybe for alternatives that are equally good, we can, again, the ebook has some interesting alternative options for visualizing Hi-C data, and I think some of them are cloud-based as well, so that could be a good alternative. Hi-C stuff pipeline is good to map and plot the heat maps. Integral is good for scaffolding. Oh, well, thank you so much, Dr. I appreciate your kind of jumping in there to answer that question. Yeah, okay, so I think there's a question around combining analysis of RNA-seq, Hi-C, and ATAC-seq, and it's possible, kind of combining that with the Arima technology. That there is multi-omics is, but kind of stacking these layers is, what a lot of our customers end up doing. I think if the question is whether we can use the same tissue for all three types of analysis, we don't have a method today to be able to do that. But certainly, taking a sample and it would be a matter of kind of breaking them up into separate experiments, and then stacking the data on top of each other would be the way to go for that one. All right. Well, in that case, I wanted to say thank you so much for your time. I hope this was an informative webinar for you. And please feel free to reach out to myself, to our wonderful team at Arima. Our scientists are here to answer any questions that you might have. I think there are some questions that we'll follow back up on, once we kind of tap into some of our bioinformatics team members to kind of answer some of those technical questions. So, look out for that, and we'll hopefully talk to you soon.